Hello students, I am here to discuss about the chapter cell, which is the biology syllabus, and in this chapter we will discuss about the animal or cell organelles. It means cell, the animal part, and in which we discuss about the organelles and their function. This is the second class in which we discuss the organelles. Previous class we discussed about the cell, their investigation means their history of investigation, as well as different theories regarding the cell. Now cell organelles, cell contain the whole part. As you know, cell is a unit of life, and this unit contain the outer covering, the middle cytoplasm and the inner nucleoplasm. So within this cytoplasm there are so many organelles, within the nucleoplasm there are so many organelles. So these organelles are able to do the activity of the function of the cell. It means the function of the cell is the function of the organelles, or collectively we can say each and every organelle what function perform is the collective function of the whole cell as well as whole body. So cell, animal cell having the outer covering, that is the plasma membrane. After that you will see these are the endoplasmic reticulum. Endoplasmic reticulum are the tube-like structure which connect the nucleus to the membrane outer covering. So they are able to form the skeleton and make the cell bulgy. Here the Golgi apparatus which is present here and there, so many Golgi apparatus are present, and they are able to do the function of storage of some substances. So later we will discuss their main functions. So third one, lysosome, present here and there in the cytoplasm, and lysosomes helps to cell digestion or intracellular digestion. After that you see ribosome, it is the protein factory of the cell and help the cell to produce protein. It means they assemble the amino acids and make the protein, that's why it is the protein factory. Now the mitochondria, you can say this is the mitochondria, so many mitochondria present inside the cell, and this mitochondria is useful for the activity of breaking of food. It means this is the powerhouse of the cell. Ribosome, protein factory of the cell. So this is the term we use for the organelles. Ribosome, protein factory, because they make the protein. Mitochondria, powerhouse, because they emit power or generate energy from the food after oxidation or breaking. Then after you will say centriole. This centriole is present here, only the part of animal cell, not found in plant cell. They are able to do the activity of division of the cell. Cell divide, and centriole help to divide the cell by pulling the chromosome or chromatids towards the opposite pole. So centriole and really the centrosome make the centriole. So these are the centrosome which make centriole after breaking, like chromosome make the chromatids after breaking. Now see here the nucleus, the main organelle which able to operate whole cell activities. So this is the nucleus which is act as a brain of the cell, and you see inside the nucleus the dense particle having substance, this is the nucleolus. Nucleolus is the center from where the nucleus is operated. Activity of nucleus is operated by nucleolus, and you see inside the nucleus or inside in the nucleoplasm there is a thread-like structure, intermixed structure, that is called chromatin thread. Chromatin thread breaks into a small thread-like structure, and after that they become thickened and very much smaller. Then after they form the chromosome, which are able to carry the characters or the features of parents to the offspring. So, chromosome hereditary characters carrying unit. So, this is the cell in which different type of organelles present in it and their function takes place. So, we will discuss one by one the plasma membrane, the endoplasmic reticulum, Golgi apparatus, lysosome, ribosome, mitochondria, centriole and nucleus. And inside the nucleus we will discuss about nucleolus and chromatin thread. So, see how the cell organelles have structures and their functions, what structure they have and what function they perform. So, first that is the plasma membrane. You know, cell divided into three parts for our easy study. So, say how they divided. So, first one, cell divided into the outer covering, that is the plasma membrane. Plasma membrane. Then after the cytoplasm. Then after the nucleoplasm. So, see here, if it is a cell, inside the cell, this is the nucleus, inside the plasma membrane and between the nuclear membrane, this is the cytoplasm. And inside the nucleus there is the liquid present is called nucleoplasm. These two meet together and form the protoplasm. Protoplasm is the life-giving substance. It means without protoplasm we can't have life. So, it is life-giving substance. So, this is the three things. So, in cell we will study one by one about the plasma membrane, then after cytoplasmic cell organelles, cytoplasmic organelles and nucleoplasmic organelles. So, in cytoplasmic organelles there is very few things, that is in this one plasma membrane, in this one the first one, that is ER, endoplasmic reticulum, second one Golgi apparatus, third one that is lysosome, fourth one that is ribosome, fifth one mitochondria, sixth one that is centrosome or centrosome. This is centrosome or centriole. Seventh one that is centriole. So, this is the cytoplasmic organelles. And nucleoplasm, inside the nucleoplasm there is nucleolus and chromatin thread. So, say this is the three heading in which we study the whole cell. Plasma membrane is the outer covering made up of two things, that is protein and lipid, fat. So protein makes the body, that is the bodybuilding things, and fat support them. So say how we discuss, how we know about the structure as well as the function of plasma membrane. See, plasma membrane, the first one, plasma membrane, it is a cell organelles present outside the animal cell, and the term plasma membrane was coined by C. Nageli, okay, C. Nageli and C. Cromer, C. Cromer. So they coined the term plasma membrane in 1885. So after that, 1855. After that, the term plasma lemma, same term given by the term plasma lemma given by a scientist named J.Q. Plowey. J.Q. Plowey. J.Q. Plowey. In 1931. Now, how it is made up of and what is its function? So, it is made up of, protein and lipid. Two layers of protein and one layer of lipid make them. So this is trilamellar structure. Trilamellar structure, which perform the activity of outer covering surrounding the cell. It is also found in plant cell under the cell wall. So in plant cell they perform the function of the same as in animal cell, but the main function of plasma membrane is the first one. It made up of the first one controls the exchange between the cell and external environment. Exchange substances between cell and outer environment. Second function of this one, it is selectively permeable membrane. It acts as selectively permeable membrane. So selectively permeable membrane means what? That membrane which select the things and then permit it to the cell or from the cell. So they mediate, it means they allow something to pass and they hinder something to not to pass. So this is the plasma membrane, which is really help the cell to make a limit. It means it also protect the cell and avoid mixing with other cell other cell. It means it separates the cell from other cells. So this is the plasma membrane which is really found on the outer surface of the cell and helps to do the function of different types like exchange of substances. They also act as circulatory permeable membrane and they protect the cell to avoid mixing of other cell organelles to it or other cells to it. Now the second one, ER. ER, its full form is endoplasmic reticulum. It is found just from the nucleus. This is the ER, from the nucleus to the plasma membrane. This is the ER. Endoplasmic reticulum is noticeable with the electron microscope. Other microscope or simple microscope is not able to see it. So ER is an extensive network of intercellular membrane bound tubes and vesicles. It occupies most of the membrane bonded tubules. So ER having two types. So on the basis of its roughness, so it is of two types. First SER, second RER. SER stands smooth endoplasmic reticulum, whereas the RER stands rough endoplasmic reticulum. So roughness and smoothness is due to the presence and absence of ribosome on it. See, this is the smooth endoplasmic reticulum, no ribosome on it, but this is the rough ER because on it the ribosome deposited, ribosome moving here and there in the cytoplasm and can deposit on it. So this is the way rough and smooth endoplasmic reticulum are present. So the function of a smooth endoplasmic reticulum is to produce lipid, means membrane formation. It acts as supporting skeletal cell framework. So they form the lipid and they form lipid with protein. They form only lipid and RER, because addition of ribosome in it, they form the lipid, the same main function of ER with the protein, because ribosome act here as the protein factory. So protein formation takes place, protein production takes place in RER, not in SER. So say what other functions they perform in the cell, because they present in the cell and support the skeletal framework. So function of ER, functions, it supports skeleton, it makes skeletal framework, skeletal framework of cell. Cell's framework made by it. Second activity, it helps to transport materials from one part of the cell to another. It means exchange of material takes place by this one. It transports material from one part to another, from one part of the cell to another. Okay. So second function is to perform its exchange. So it helps to synthesis protein as well as lipid. You know, here protein lipid synthesis takes place by this one. Next function, it helps in the formation of Golgi apparatus, lysosome and vacuoles. Main function is this. So it helps in the formation of Golgi body, lysosome and vacuoles So this is the function of endoplasmic reticulum. So we discussed today ER and plasma membrane. Next day we will discuss about the other cell organelles. Okay, thank you.